GEMSA staining procedure. G-bending, which is also called as GEMSA staining, is obtained after the digestion of the chromosomes with the enzyme. The name of the enzyme is trypsin. What is a GEMSA stain? GEMSA stain is a mixture of methylene blue, eosin, as well as azure B. So it is a mixture of these three things. When we combine them, so it forms in the form of GEMSA stain. It is specific dye, or it is a specific stain which usually binds to the phosphate groups which are present at adenine and thymine. So when we use GEMSA stain, it binds with the phosphate groups of adenine and thymine and gives them the dark color. GEMSA stain produces light bands and dark bands. They are also called as dark G bands and light G bands. Dark regions are those regions which are heterochromatin. They are late replicating and they are AT rich. While on the other hand, light regions are those regions which are euchromatin. They are early replicating and they are GC rich regions, guanine and cytosine. In this diagram, we can see that a chromosome has been stained with the help of GEMSA, and after the staining of the chromosome, we can see that there are some dark bands and there are some light bands. Like here we can see that this is dark band and here is light band. Again here is dark band. Same is here in case of Q arm, where we can see this is dark band, here is light band, this one is dark band and this one is light band. So with the help of a GEMSA stain, when we stain the chromosomes, then as a result of the staining, we can see that some of the fragments of the DNA, they are dark and some of them, they are light. This is the procedure that how chromosomes are stained with the help of GEMSA. So this is the initial procedure how we prepare the slides for the chromosomes. So the first step is the collection of the bloods, then cell culturing, that is the second step. Once we collect the blood samples, then we culture the cells, those cells which are present in the blood, that can be red blood cells, that can be white blood cells. So that is the culturing of the cells which are present in the blood. And the third step is cell division at the metaphase. When we culture the cells, so there is a process of division of the cells. Cell divide into two daughter cells. So this cell division at metaphase is stopped. The reason is that at this stage, chromosomes, they are dense and they are more visible. So that is the reason that this cell division is stopped at the metaphase of the mitosis. Once cell division is stopped, then hypotonic treatment is performed both for the red blood cells and for the white blood cells. During the next step, there is fixation and slides preparation, then slide dehydration, so that there is a removal of any kind of the water. Then we treat those slides with an enzyme. The name of the enzyme is trypsin. And then we stain those slides to see the bending pattern with the help of GEMSA. So during the first step, we prepare the slides by taking the blood samples. So this is the procedure for preparing the slides. Once we have prepared the slides, the whole process is like in this pattern. In the case of peripheral blood sample is added to small volume of nutrient medium. There is a nutrient medium on which the blood sample is added. This nutrient medium contains a chemical. The name of the chemical is phytohemagglutinin. So this chemical stimulates the T lymphocyte to divide during the preparation of the slides. Cells are cultured under sterile condition at about 37 degrees centigrade for 3 days. During they divide, the cells and the chromosomes, they divide, and once they have been divided in a sufficient amount, then a chemical is added. The name of the chemical is colchicine. Colchicine is a chemical that has extremely useful property for preventing the formation of the spindle fibers. So this is the stage of metaphase. When the chromosomes, they are at metaphase in some of the cells, then there is addition of the chemical. The name of the chemical is colchicine. Once we add the colchicine, it stops the formation of the spindles. So once spindles, they are stopped, their formation is stopped, then the cell goes in the resting phase, thereby resting the cell division at metaphase. The time chromosomes, this is the time at the stage of metaphase when chromosomes, they are most visible, they are most thick, and we can easily view them under the microscope. Hypotonic saline solution is then added, which results the cell to lyse, because we have stopped the cell division with the help of colchicine. Once we have stopped, then we add the hypotonic saline solution to lyse the cells or to burst the cells, so that the chromosomes, they come out of the cells or from the nucleus. And once they come out of the cells or nucleus, we fix them mounted on the slide, and now they are ready for the analysis. This complete process is called as GEM731 staining. So these are the bands we can view once we use GEM731 stain for the studying of the chromosomes. So in case of chromosome 1, if we take just the example of chromosome 1, we can see that there are different bands. This is dark band, then light band, then dark band, light band. Same is with all the chromosomes. We can see dark band and light bands. So then what is GEM731 staining? GEM731 staining is a procedure, or GEM731 is a stain or chemical which is used to stain the chromosomes. Once we stain the chromosomes, we can see chromosomes in the form of dark bands and light bands. Dark regions, they are heterochromatin and they are AT rich region, and these are those regions which replicate late during the cell cycle. While on the other hand, light bands are those bands which are euchromatin, they are GC rich region, and they early replicate during the process of cell cycle.